So we have a very interesting application today. We have an application where we're doing some characterization of size and shape of stem cells. So stem cells are very unique in that, actually, they're not unique. They're undifferentiated is actually more accurate. Stem cells are is a very hot topic these days. It's an area of interest because stem cells are cells that are undifferentiated. They are cells that actually can eventually differentiate themselves into different areas. There's a large amount of research going into stem cells and how they can actually help with therapeutics and so forth. So it was very interesting and very pleasant to actually have the opportunity to look at some stem cell information, and that's what we have here. We have some stem cell information. We did some particle size analysis of it. So to go over briefly what we're looking at a particle size and shape analysis of some stem cells. We're done with the actual information. The Particle Insight is an instrument that gives you particle size and shape. We have histograms for all the measured particles. So we measured a total of 52,000 particles, and we have all the different shape measures that are here. I collected all the shape measures because we weren't really sure what we were looking at. It's always good to capture all the shape information in real time and then try to see what we have after the fact. Now, with stem cells, we try to look. I'm assuming based on some of my background, we're looking at some of the size information. We're looking at some of the opacity information, whether they're alive or dead, even though this is really not a viability analysis. And I also look at circularity. There could be some information on the shape of the particles that could be used for research. Maybe as the stem cells become more elongated, they're moving toward a process of dying out or apoptosis, or they're moving toward a direction toward differentiating themselves into a specific type of cell. So all the information that we have will be stored here. The Particle Insight stores all the data for all the particles as well as the thumbnails. The thumbnails is always very unique in that it gives us the information to look at visually to see what we can determine. So the three shape measures that I picked were diameter, because size is always something that's very unique to look at and very common to look at. So these had a number-based distribution of about a mean about 13 microns. I like to look at circularity to see how round these particles are, and then opacity to see how dark they are if there were subpopulations in there. We also have the system performance data, which actually shows that we had in this sample about 1.8 million particles per milliliter measured. So it was semi-concentrated, not too much, not too low. When we look at the actual data results, generally what I tend to do is I go in and I'll go into the post-run processing of the Particle Insight, and I will look to see what the overall view of the different particles were. By doing that, I get an idea of just basically what I'm looking at. So I'll go ahead and I'll press the thumbnail images, and then I'll load up all the thumbnails. This is just all the thumbnails, which might be a little bit overwhelming, but we'll just open all the thumbnails that were captured for the all 50,000 particles. So now the thumbnails open up, and I could scroll through them. I can obviously see that I had some highly concentrated sample that can actually increase my cluster rejection to minimize that. But I do also see some very unique particles that do look like they're longer or fused. Other observations that I make is that there are some lighter particles and there's some darker particles. There's some elongated ones. So that's something that might be of use to a researcher. Also, as I scroll through, I'm definitely seeing some parts of particles, some parts of cells that look very different shapes. Maybe those are cells that are going through the death process, the dying process. I also noticed light and dark, and also as I scroll through, there are some particles that now they start looking smaller as well. So that's some information that's actually very useful. Now keep in mind, the Particle Insight, besides being a size and shape analyzer, we also do enumeration, so we can tell you not only the concentration, but we can tell you the percentage of small particles or dark particles in the entire population, if that's something that's important to know. So this is way too much information. We have just scrolling through these thumbnails. It doesn't quantify anything for us. It's just qualitative information. What I tend to do once I look at all the different thumbnails and I look to see what, let's say, all these stem cells look like, I'll go in and I'll start making some assumptions. So the first assumption I will make is I will look at let's say the correlation. So one of the things I'll do first is I'll try to make a general assumption. Let's say I want to determine and see if the cells as they change size, do they become darker, and I can do a correlation plot and see if there's any outliers. I can see right now, even though it's laying out all the particles, I can see that this kind of correlates with the ECA diameter plot, where we're seeing you know, population that goes up to about below 2 microns, and then this main population at about the 13 micron range. And I can see they're all centered pretty much close to the same opacity. So this opacity really, this comparison of size versus opacity doesn't really tell me a whole lot. What I can do, again, I'm not the educated eye here. I can look to see maybe if as they're changing size, are they getting more or less round. So I can look at circularity, and I can do a correlation plot there. Here I do see some outliers, so this might be of interest. So if this would be of interest to a person studying stem cells, they may be interested in knowing that looks like the circularity of the main population is pretty round, but that there is definitely a population that's a lot smaller. So it looks like these small particles down here in the 2 micron range seem to be a lot smaller, but then there's also some less round ones here. Some of the round ones here can actually be some of those agglomerates, you know, the ones that were stuck together. And we can easily go in and look at all the thumbnails and get an attributable cause to what all these dots are. So this is one of the nice tools of the Particle Insight. I call it the finding the needle in the haystack. So this is the rare event detector. So this is where I can look, you know, in an ocean of 50,000 particles. If I wanted to find out why I have 1 or 2 particles that had an extremely high circularity and were very small, I can go in and look to see what those particles are. So it would be impossible to do that just going through a wave of 50,000 images. But when you do this rare event detection feature, you can actually look for those rare events. So that's one nice feature with the Particle Insight. The other nice feature that we can look at with the Particle Insight, and I already talked about concentration, is particle classification. So particle classification is something that we can go in and we can actually look at specific areas of the population independent of other things. What do I mean by that? So in this case, I went ahead and I created a group of different classes. I called some small round cells, large round cells, long cells, dark cells, light cells, and then unknown. So what I did is I went in and I created parameters for all these, and then I actually could see what count there is for all these different particles. Not only can I do that, but for example, let's say I wanted to find out what the round, well, no, let's look what the round small cells. I can actually look at what the round small cells. I could get all the statistics. So I can actually look at only the round small cells. I can look at all the statistics just of the round small cells, ignoring all the other particles. So this is the equivalent of actually, with a click of a button, doing a full shape analysis and size analysis with 30 shape measures of just the particles that are round and small. So we can look at the different statistics based on that. We can actually look at and there they are. So these are all the round small cells. You can see I cut it off at a certain size, but it also affects all the different data on the other histogram. So again, this is just the small round cells. I can also go in and just say, okay, well, let's say the large round cells, I only want to look at the thumbnail images of the large round cells. So I can actually, and not only do you have the quantification, exactly how many there were found, what the percentage of total was, what their mean size or standard deviation, all the statistics here, but I could also validate that looking at the different thumbnails. Okay, so here we have what I took a stab in the dark here, and I took a shot saying that these are the larger round ones. So I can also keep on, I can also keep going and looking at, let's say, out of the large round cells, which are the dark ones and which are the light ones. I can do that as well. or I could say out of the large round cells, which ones are smoother. So there's many different ways that you can actually look at this data and have it give you quantifiable information. So again, this is another tool where you can actually quantify what specific part of the population you're looking for. Okay, so that's it. That's all I have for today. I'll post this online for people to look at, and if there's any questions, please contact us at your convenience. Thank you.